Updating the Language and Conceptual Framework of Evolutionary Theory. How history and language have caused confusion that extends beyond the modern synthesis to neo-Darwinian theory. In his clear and insightful 2015 synthesis of ideas, put forward in his earlier books and lectures, Denis Noble pointed out how the web of interpretation that represents the whole conceptual scheme of neo-Darwinism has created significant difficulties for evolutionary theory. Each concept, simile, or metaphor reinforces the overall mindset until it is almost impossible to stand outside it and appreciate how beguiling it is. As the modern synthesis has dominated biological science for over half a century, its viewpoint is now so embedded in the scientific literature, including standard school and university textbooks, that many biological scientists may not recognize its conceptual nature, let alone question incoherencies or identify flaws. Noble correctly points out that many of the problems with the modern synthesis arise from the way that neo-Darwinism represents the information, rather than the empirical findings per se. These forms of representation have been responsible for and express the way in which 20th century biology has most frequently been interpreted. The concepts, therefore, form a biased interpretive veneer that can hide those discoveries in a web of interpretation. No single word has caused greater problems for evolutionary theory than the word gene. To understand this, we will need to look more closely at the gap between revolutionary empirical discoveries about the nature of the gene and the entrenched conceptual framework of the modern synthesis. Noble provides an insightful analysis of the conceptual and semantic dichotomy between the Mendelian concept of the gene and the term gene J to describe the conceptual dichotomy it has created. The word gene was introduced by Johansen, 1909, but the concept had already existed since Mendel's experiments on plant hybrids, published in 1866, see Drury and Bateson, 1901, and was based on the silent assumption that was made almost universally that there is a one-to-one relation between genetic factor, gene, and character, Mayer, 1982. Of course, no one now thinks that there is a simple one-to-one relation, but the language of direct causation has been retained. I will call this definition of a gene, gene J, to signify Johansen's, but essentially also Mendel's meaning. The gene J definition subtly implies that there is a one-to-one relationship between a genetic factor and a specific visible characteristic of an individual, consistent with the visible correlation between the presence of a specific genetic allele and the visible phenotype, character, that is observed when that allele is present. Gene J referred to the cause of a specific inheritable phenotype characteristic, trait, such as eye, hair, skin color, body shape and mass, number of legs, arms, wings, to which we could perhaps add more complex traits such as intelligence, personality, and sexuality. Having summarized some of the key empirical discoveries of molecular biology or molecular genetics from the 1960s, we can now return to Denis Noble's discussion of the conceptual baggage of the modern synthesis and neo-Darwinism. The empirical discoveries of molecular genetics revolutionized our understanding of the nature of the gene, and Noble draws a distinction between the older definition of gene J and a new one. Following the discovery that DNA forms templates for proteins, the definition shifted to locatable DNA sequences with identifiable beginnings and endings. Complexity was added through the discovery of regulatory elements, essentially switches, but the basic cause of phenotype characteristics was still thought to be the DNA sequence, as that forms the template to determine which protein is made, which in turn interacts with the rest of the organism to produce the phenotype. I will call this definition of a gene, gene M. The M subscript in gene M represents a molecular definition of the gene. The problem, however, is that the conceptual framework of the modern synthesis, which was closely tied to the Mendelian genetics of the Drosophila experiments, continued to associate genetic variation with the random mutations described by Julian Huxley.